Today we're diving into one of the most fascinating topics in human biology: how we sense the world around us. Every sight, sound, taste, and touch we experience begins with a single process: stimulus detection by sensory receptors. Think of this chapter as the biology of perception, where physics meets physiology and turns raw data into the human experience. Information from both the external world and inside our bodies travels to the brain as tiny electrical impulses. These come from sensory receptors specialized to detect change, like pressure, temperature, or even light. Once those receptors are stimulated, they send action potentials through sensory neurons to the brain, where the information becomes perception. A stimulus is any detectable change coming from either heat, sound, light, or even chemical. Receptors convert that stimulus energy into another form, often an electrical signal. This conversion process is called transduction. Some receptors release neurotransmitters to signal nearby neurons. When the brain becomes consciously aware of that input, we call it sensation. When it interprets the meaning of it, like recognizing the smell of pizza, it's called perception. Different receptors are tuned to different kinds of energy. Mechanoreceptors detect pressure or sound. Thermoreceptors respond to temperature. Chemoreceptors detect chemicals for taste and smell. Photoreceptors respond to light, and pain receptors alert us to harm. Each one translates its type of stimulus into a form of nervous system where it can understand, which is known as an electrical message. Once a signal reaches the brain, it's interpreted based on where it came from and how often the neuron fires. Stronger stimuli don't make stronger impulses, they make more of them, or they fire faster. This is how the brain distinguishes gentle touch from firm pressure or a quiet whisper from a loud noise. The pattern and frequency of firing gives each sensation its intensity and meaning. Not every receptor keeps firing their entire time a stimulus is present. Sensory adaptation happens when certain receptors stop sending signals even though the stimulus continues. For example, you notice a smell when you first walk into a room, but after a few minutes, you stop noticing it. That's your nervous system tuning out unnecessary background information so you can focus on what's new or important. However, receptors for pain or joint positions don't adapt because that information is critical for protection and movement. We divide sensory input into somatic sensation and special senses. Somatic sensations come from the receptors all over your body, like those for touch, temperature, or pain. Special senses are localized in specific organs, such as your eyes, ears, nose, or tongue. Together, they give us a complete picture of our environment, allowing us to interact with the world and maintain balance and awareness. Somatic receptors detect things like temperature, pressure, vibrations, pain, and body positions. Once activated, they send signals to the primary somatosensory cortex in the parietal lobes, which identify the types and locations of the sensation. That information is then sent to the motor cortex in the frontal lobe so we can respond, like pulling our hands away from hot surface. This back and forth communication keeps sensory input and motor output tightly connected. Our skin is packed with specialized receptors for different types of touch. Free nerve endings detect pain and temperature. Merkel discs and Meissner corpuscles sense light touch, while Ruffini endings detect continuous pressure, and Pacinian corpuscles respond to keep pressure or vibration. These work together to help us detect everything from feather brushings on the skin to the weight of our backpacks. Mechanoreceptors aren't just in the skin, they're also deep in muscles, tendons, and joints. Muscle spindles monitor how stretched a muscle is, while tendon receptors detect tension. Joint receptors tell the brain the position of our limbs. This sense is also called proprioception, which allows us to move without constantly looking at our body. These receptors even play a protective role. If tension gets too high, they trigger reflexes that prevent muscle damage. Thermoreceptors near the surface of the skin monitor the external environment, giving us the feedback about warmth or cold. These receptors adapt quickly, like when you first jump into the pool and then just after a minute or two. In contrast, thermoreceptors deep in the body, such as the thoracic or abdominal organs, monitor core body temperature and adapt slowly. This helps the brain regulate internal temperatures and maintain homeostasis. Pain receptors, or nociceptors, are free nerve endings that respond to potential harmful stimuli, There are two main types: fast pain, which is sharp and immediate, and slow pain, which is dull and lasts longer. Slow pain often comes from the internal organs, which is why it can feel diffuse or even hard to locate. Sometimes pain is referred, meaning it is felt in a different area from its source, like shoulder pain during a heart attack. Pain receptors don't adapt because they serve as a constant warning system. Taste begins with food molecules dissolved in saliva that bind to chemoreceptors on taste buds. Each taste bud contains several receptor cells sensitive to specific chemicals. Humans can detect five main taste categories: sweet, salty, sour, bitter, and umami, which is a savory flavor. Most of our taste buds are around the edges of the back of our tongue. These signals travel to the brain, which combines them with the information from smell to create the full experience of flavor. Our sense of smell, or olfaction, comes from the receptors on the upper nasal cavity. Each receptor binds to specific odor molecules called odorants. Humans can detect over 400 types of odorants and distinguish thousands of different scents. Smell and taste work closely together. When you chew your food, volatile chemicals travel up your nasal cavity, enhancing flavor. That's why food tastes bland when you have a cold or a stuffy nose. Sound is simply waves of compressed air traveling through space. Our ears convert those pressure waves into electrical signals using mechanoreceptors. The loudness of the sound depends on the amplitude of the wave, while pitch depends on the frequency. These vibrations eventually reach the inner ear, where specialized hair cells turn the movement of sound waves into action potentials the brain interprets as sound. This process is remarkably precise, allowing us to enjoy music, recognize voice, and detect danger. Hearing begins when sound waves enter the outer ear. The pinna, which is the visible portion of the ear, acts as the funnel that captures and directs sound into the auditory canal. These waves travel inward. They strike the tympanic membrane or eardrum, causing it to vibrate back and forth. These vibrations are the first step in converting sound energy from air pressure into mechanical movements that the ear can actually process further. The middle ear is air-filled chambers that contains the malleus, the incus, and the stapes. These three bones transmit and amplify vibrations from the eardrum to the oval window in the inner ear. The auditory tube, or eustachian tube, connects to the throat and helps equalize air pressure on both sides of the eardrum, which is important for normal hearing. Inside the cochlea, sound vibrations are transformed into electrical impulses. The cochlea is the coiled, fluid-filled structure that contains the organ of Corti. This organ houses hair cells, which are the mechanoreceptors for hearing. As sound waves move through the cochlear fluid, they bend the tiny hairs on these cells. Bending opens ion channels that trigger action potentials, which then travels through the auditory nerve towards the brain for interpretation. Different frequencies of sound stimulate specific regions of the cochlea. High-frequency sound vibrations vibrate hair near the base, close to the oval window, while low-frequency sounds activate hair cells on the apex of the cochlea. The bending of hair cells creates a pattern of electrical impulses that represents both the pitch and intensity of the sound. These signals are carried to the brain through the vestibular cochlear nerve for processing. The vestibular apparatus of the inner ear includes three semicircular canals and a central chamber called a vestibule. The semicircular canals detect rotational movements when fluid inside them shifts and bends sensory hair cells. The vestibule, containing the utricle and the saccule, detect gravity and linear acceleration through tiny calcium crystals called otoliths. Together, these structures send constant signals to the brain about head movement and body position, allowing coordination and balance. Vision depends on light entering the eye and being converted into nerve impulses. Light first passes through the cornea and the lens, which work together to focus into the retina. The retina contains photoreceptors that detect light and send information through the optic nerve to the brain. The vitreous humor helps maintain the shape of the eye and ensures that light is properly directed towards the back of the eye, where perception begins. The iris, which adjusts the diameter of the pupil to control the amount of light entering the eye, is made up of smooth muscle. In bright light, the pupil constricts, while in dim light it dilates. The lens changes curvature in a process called accommodation to focus on near or distant objects. The ciliary muscle surrounding the lens controls this process. As we age, the lens becomes less flexible, reducing the ability to focus on nearby objects, a condition known as presbyopia. The shape of the eye determines where light focuses on the retina. In a normal eye, light converges precisely on the retina, producing a clear image. In myopia or nearsightedness, the eyeball is too long, causing distant objects to appear blurry. In hyperopia or farsightedness, the eyeball is too short, so near objects focus behind the retina. Astigmatisms result from uneven curvature of the cornea or lens, distorting vision in all directions. Corrective lenses adjust the path of light so it properly focuses on the retina. The retina is made up of multiple layers of cells that transform light into electrical signals. The outermost layer absorbs excessive light, preventing reflection. The middle layer contains rods and cones which respond to light and color. These photoreceptors communicate with bipolar cells, which in turn connect to ganglion cells. The axons of ganglion cells merge to form the optic nerve, which transmits visual information to the brain's occipital lobe for processing. Rods and cones differ in sensitivity and function. Rods are highly sensitive to light and enable vision in dim environments, but they do not detect color. Cones function in bright light and provide sharp color vision. There are 3 types of cones, red, green, and blue, each tuned to different wavelengths of light. The brain interprets color by comparing the combined activity of these cone types, producing the full spectrum of color perception. Rods contain a light-sensitive pigment called rhopsin, which is much more sensitive to light than the pigments in cones. This allows rods to function well in dim environments, such as at night or in dark rooms. Because rods do not detect color, vision under low light is limited to shades of gray. The density of rods is highest away from the center of the retina, which is why objects are often easier to see at night when you look slightly to the side of them, rather than directly at them. Cones are concentrated near the fovea, the region of the retina responsible for sharp, detailed vision. Each type of cone contains a specific pigment that absorbs red, green, or blue wavelengths of light. The brain compares input from all 3 types of cones to produce color vision. Cones require more light to be activated than rods, which is why color vision fades in dim light and returns in bright conditions. Visual adaptations occur when the eyes adjust to the changes in light levels. Moving from bright areas to dark ones cause rhopsin in rods to regenerate slowly, leading to temporary poor night vision until it builds up again. This process can take several minutes. When moving from dark to bright conditions, rhopsin breaks down quickly, and the sensitivity of the rods decreases while cones take over. The iris also contributes by adjusting pupil size to regulate light entering the eye. Hearing loss can result from problems in different parts of the ear. Nerve deafness occurs when hair cells in the cochlea are damaged, often from prolonged exposure to loud sounds. Conduction deafness happens when vibrations cannot pass efficiently through the middle ear due to the damage of stiffness in the ossicles or tympanic membrane. Hearing aids can amplify sound for conditions problems, while cochlear implants can help replace the function of the damaged hair cells in nerve deafness. Otitis media is a common middle ear infection, particularly in infants and young children. It often develops from an upper respiratory infection spread through the eustachian tubes to the middle ear. The infection can cause fluid buildup, inflammation, and temporary hearing loss. If caused by bacteria, it's treated with antibiotics. In recent cases, small tubes may be inserted through the tympanic membrane to help fluid drain and prevent pressure buildup. Many-ear syndrome is a chronic disorder of the inner ear that affects both hearing and balance. It is thought to be related to fluid buildup in the inner ear. Symptoms include episodes of vertigo, nausea, ringing in the ear, and progressive hearing loss. Severe attacks can disrupt balance to the point that the standing or walking becomes difficult. Treatment may include medications for dizziness or surgery to relieve fluid pressure. Retinal detachment occurs when the retina separates from the underlying tissues, leading to blurred vision, flashes of light, or loss of peripheral vision. Prompt surgical repair can prevent permanent damage. Cataracts occurs when the lens becomes cloudy, reducing light transmission and blurry vision. They are often age-related or linked to diabetes and can be treated by replacing the clouded lens with an artificial one. Glaucoma results from the increased pressure in the eye due to poor drainage of aqueous humor, which can damage the optic nerve if untreated. Age-related macular degeneration, or AMD, causes deteriorations of the macula, the part of the retina responsible for sharp central vision. This leads to loss of details and difficulty seeing directly ahead. It is the leading cause of blindness in older adults. Color blindness results from the absence or malfunction of one or more types of cones. The most common form is red-green color blindness, which is usually inherited and more frequent in males.